This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 278. Recorded on December 15th, 2022. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michelle Swanson.
Hello, nice to see you both.
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone, where we have a balmy 71 or 20 degrees centigrade.
Wow.
Oh, that southern weather, huh?
Yes.
How about that? Well, this is probably our second to last episode of the year, I'd guess. We do have a potential for the 29th, so I won't say goodbye to everyone yet. Rather, we will jump right into our papers for today. We're going to start with Michael's in a snippet.
Well, something that has been on the news over these last 2 weeks in our run-up to the holidays has been the discussion about depression. Now, depression is one of the most common mental health disorders experienced worldwide, with an average lifetime prevalence of between 11 and 15%. Now, the prevalence of depression has doubled, and in some countries, even tripled during the COVID pandemic.
That's depressing.
Yeah. Plus, the two papers that we are commending for your review, and they're both open access, and they've been published the week of December 6th in Nature Communications, have been making their rounds on the news and in publications like the Wall Street Journal. The first paper and there are many authors, and they're principally from Erasmus Medical Center in Rotterdam in the Netherlands, is Rajab Zadeh and his colleagues, and it's entitled A Gut Microbiome-Wide Association Study of Depressive Symptoms. Now, as an introduction, I've been teaching a course in evidence-based practice this semester to our D1 dental students. They call it evidence-based dentistry, but it's really evidence-based practice. And they have been learning about the different types of clinical studies. And I've been stressing the importance to them of understanding the type of study authors conduct and how that study design can help the authors get at the question they are asking, which in this case is whether or not microbes, specifically gut microorganisms, are involved in the pathophysiology of clinical depression. So we need to start with the definition so that you, the listeners, know what the authors are investigating. Operationally, this is an easy one, as the study design is embedded in their title. Namely, it's an association study, but with a twist. Normally, genome-wide association studies are analysis of genes or genetic alleles. But in this case, we are looking at amplicon sequence variants, or ASVs, which in this particular circumstance is really nothing more than a targeted next-generation sequence analysis specifically to the 16S ribosomal RNA gene as the molecular signature, reporting the microbial census from stool samples rather than looking at genetic alleles of the study subjects that we typically see in genome-wide association studies. Now, you recall that the question that they're asking is, are microbes that humans are harboring, are there significant variations in the microbes, or the genes, if you will, that the microbes possess that specifically genetically contribute to the pathophysiology or the development or the pathophysiology of the condition that they're investigating, namely depression? And the reason they did this study is previously association studies have been conducted looking at depression, and they looked specifically at the human genome. And these large genome-wide association studies of depression found low to moderate heritability and a very small effect of human genetic variants, where the odds ratios for those types of studies was approximately less than 1. Recall that an odds ratio is nothing more than a probability. It's a measure of an association between an exposure. In this particular circumstance that we're going to be looking at is whether or not a particular group of microorganisms are responsible or associated with the phenotype that they're scoring. In this case, it's depression. So to give you a statistical cheat sheet, if you will, if your odds ratio is about 1, the exposure or the presence of the microbe or the microbial community profile that you're characterizing does not impact the odds of having depression. If your odds ratio is greater than 1, your exposure to a particular microbe or its presence in a microbial community will be associated with a higher likelihood of the measurable outcome, in this case, clinical depression. And if the odds ratio is less than 1, exposure is associated with lower odds of displaying the clinical phenotype of depression. Now, since there was not a very strong human gene association with depression, the mental health community is urgently trying to identify both molecular and biological mechanisms underlying depression so that they can get people help sooner. And the gut microbiome is obviously a novel area of interest because there is that emerging concept of bidirectional signaling between our gut microorganisms and our brain. And this, of course, is a very active field of research. We have talked about such signaling in the past on TWiM when we discussed the link between microbes and autism, which was TWiM 131, Mice Behaving Badly. And that was in 2016, Vincent. And I'll put a link to it in the show notes so you can see what that episode was about. But recently, that link between autism and microbes was reviewed in an open source review article, Frontiers in Cellular and Infectious Microbiology. And I'll put a link to that open access article as well in the show notes. And that was reviewed in July of this year. And if you look at that review article, their first figure has a really neat schematic of what they think is going on between the microbiota in our gut and how it's interacting with our brain. So I'll let you guys look at that at your leisure since it's an open access and it's going to be in the show notes. Other recent studies have also suggested that our gut microbes can modulate this access via multiple mechanisms, including alterations in the population of microorganisms that are present, or this production of microbial neuroactive metabolites. And thus far, these associations have been mostly reported for animal models, and the human studies are a bit limited in the number of study subjects that they have had in them. And as recently as 2019, there was a paper in Nature Microbiology, also dumped into the show notes, that you can get access to pretty easily, that is looking at microorganisms. And they talk about butyrate producing fecal bacterium and coprococcus species that are typically associated with higher quality of life indicators, which is code for lower rates of depression. So,
and Michael, before you go too far, like as you pointed out, humans are complicated. It's harder to do well-controlled experiments. But there are some really beautiful classic examples in the literature of how microbes change the behavior of their host. And the one that I love is, because it's so easy to visualize, is there's a parasite, Toxoplasma gondii, that can only mate when it's within a cat. So not in soil. It's got to find its mate while it's in a cat. And they found that when rats or mice are infected with Toxoplasma gondii, the rodents lose their aversion to the smell of cat urine. So instead of like avoiding areas where there are cats, the white mice that are infected with toxo will wander about, get eaten, and now they have a chance to mate.
That is indeed the case. And that's what's so wild about this. And it's why the mental health community is looking to our gut microflora and microorganisms in general to see whether or not this can actually help them try to figure this out. So in the first paper, Rajabdaza and colleagues are going after an association study of depressive symptoms where they investigate the relationship of fecal microbiome diversity and the composition and our composition of microorganisms with depressive symptoms. And they looked at approximately 1,000 study participants from the Rotterdam study cohort, and then they validated their observations using another large cohort of individuals, namely the Amsterdam Helios cohort that had 1,539 study subjects. And they were able to control for lifestyle factors and medication use. And they also then conducted a Mendelian randomization analysis, which to the non-geneticist is a method of measured variation of genes of known function to examine the causal effect of a modifiable exposure on disease. And the modifiable exposure is whether or not you have a particular microbe or a particular community of microbes present. And so their overall goal is to elucidate, if you will, a causal relationship. But human beings are complicated, and causality is really hard to prove. And you almost have to be a jailhouse lawyer to prove it beyond a reasonable doubt, to identify the microbiota that are associated with major depression. Yet, this study was big enough that these authors learned that there was indeed an association of 13 microbial taxa, including the genera Agathella, Subdulidium granulum. These have too many syllables. Coprococcus, Celiomonas, Lancoclostridium, Hungatelia, Ruminococciaceae, Lacnospiraceae, Eubacterium ventrosum, Ruminococcus gavarelli group, and the family Ruminococci in general. And they were associated with depressive symptoms. It also should be pointed out that these bacteria are known to be involved in the synthesis of reactive chemicals like glutamate and butyrate and serotonin and gamma aminobutyric acid, or GABA, which are key neurotransmitters for depression. Of the three microbial families that lit up the dashboard, so to speak, with significantly being associated with depressive symptoms, only the Ruminococcus family was significantly associated with these depressive symptoms in the Rotterdam cohort. And while the Ruminococci in general were associated with the depressive symptoms in the Helios cohort, they did another comparison to ask if there was really any difference between the Rotterdam cohort and the Helios cohort. And they conducted a random forest analysis which, if you've forgotten your statistics, is a supervised machine learning algorithm that grows and combines multiple decision trees to create a forest. And in this random forest analysis, the Rotterdam was used as their training cohort, and the Helios was the testing cohort. And it again reveals that Rumicocciaceae, a specific amplicon sequence variant that they characterized as UCG005, was the most important genus in this particular analysis for predicting these depressive symptoms. When they further looked at the Mendelian randomization analysis, they were able to identify a causal link between major depression and this one that is called ergothella. And I found that to be fascinating. So as your population gets bigger, the power of the genetics gets a little bit more robust in these association studies. Among the 87 depression-associated single nucleotide polymorphisms, or SNPs, SNP7 had the significant association was observed, and it was associated with the genus Acidaminococcus after correction for multiple testing. And there was no significant association observed for MDD or GRS, and they then dump us to the supplementary data. So in this first paper, to summarize, they have identified several bacterial genera that might influence depression in humans. They were
or vice versa.
Or vice versa.
It's just an association.
It's just an association. They were able to confirm this association to Ergothella, Coprococcus, Subdola granulum, and the family Rumicocciaceae. And they were able to identify specific bacteria, including the Cellumonas, the Lanclostridium, the Hungatella, the Ruminococcus, the Subdolineal granulum, the Lactospiraceae, this Eubacterium species, and another Ruminococcus group. Additionally, they again point out that these bacteria are involved in the synthesis of these short-chain fatty acids like glutamate and butyrate and serotonin and GABA, which are, again, the key neurotransmitters relevant for depression. The next paper that this group is responsible for, and the one that I found to be a bit more interesting, is entitled "The Gut Microbiota and Depressive Symptoms Across Ethnic Groups." And this was by the second author of the first paper, becomes the first author. So this is Bosch and colleagues. And the primary aim of this second paper is to identify which taxonomic features of the gut microbiota are linked to depressive symptoms levels. And they measured this by using the PHQ-9 instrument, which is
the what?
I'm going to get to that. The PHQ-9 instrument is a mental health survey that can function as a screening tool. And this is effectively what scores the depressive phenotype. It's designed to aid in diagnosis and as a symptom tracking tool that can help track a patient's overall depression severity, as well as track the improvement of specific symptoms upon treatment. So this is quite often used in the mental health community to score how well your medications are alleviating your symptoms, because oftentimes the medications that are used are, everything looks like a nail, and most of the medications are hammers. And so they're throwing these medications at folks. It's also one of the limitations that the authors point out of this study is that they're using this PHQ-9 instrument in that it does not provide a clinical diagnosis of depression. It's not like a blood test where you get hard, quantitative value. But these assessment approaches, like these survey instruments in the PQ-9, tend to be highly correlative. And so, nevertheless, I think despite the limitations, you'll find the results from this study to be quite compelling. Again, this investigation involved the largest study cohort to date, with over 3,200 study subjects examining microbiome depression associations. And it was the first study to consider ethnicity as a potentially relevant factor in this association. And what these authors learned is that there were consistent associations between the gut microbiota and depressive symptoms levels, and they were able to confirm this at multiple levels of analyses, ranging from global parameters of microbiota diversity as measured by the two principal ways of looking at things: alpha diversity, which is, if you will, your microbiome measured longitudinally, and beta diversity, which would be the microbiome of Michael, Michelle, and Vincent, if we were to compare ourselves to the relative abundance of specific taxa. Now, these associations withstood a broad range of sociodemographic, behavioral, and medical covariates. Their analyses further revealed that these associations were largely invariant across ethnic groups, notwithstanding substantial ethnic differences in both depressive symptoms levels and the composition of the gut microbiota. Moreover,
that's especially striking because in general, people of different ethnic backgrounds maybe have slightly different diets, which could also change their microbiome.
And they were able, and I'll just sort of jump here for the sake of time, what they learned is while alpha diversity and depression could be correlated, it didn't stand up to the rigors of a covariable that they threw in to their analysis. And that covariant was a behavior trait known as neuroticism. And neuroticism is whether or not you're neurotic. It's whether or not you experience negative effects, including anger, anxiety, self-consciousness, irritability, emotional instability, and depression. If you had this global trait, and again, it's a survey tool that you tick off the box. You feel anger? Do you feel anxious? And that will then score whether or not you're neurotic. If you're neurotic, alpha diversity no longer is significantly predictive for depressive symptoms. And they adjusted that in each of their 3 linear regression models, which again gets to Michelle's point of accounting for differences in diet.
So it's kind of a specificity control, if you were.
Yes. So
it is specific to depression, but whether it's causal or consequence.
It's only an association. As you point out, you don't know which way it's going. And so this study actually gives the microbiologists, and why I picked the study as my favorite, it gives us, the microbial biology community and the neuroscientists, something to think about as we begin to design our experiments. And upon further refining their model, they found 23 amplicon sequence variants, identifying at least 15 genera that may remain significantly associated with depression scores. Now, there was a negative association with the abundant genus Coprococcus. So if you have Coprococcus in your gut, you're not going to be depressed. And that was confirmed with 2 independent population cohorts and the Rotterdam studies. Now, this genus, the Coprococcus, harbors many butyrate-producing species and has been ascribed to anti-inflammatory properties, which have been inversely linked to depression. Additionally, and of interest to my dental students, their analyses showed a positive association. So that means if you have this bug, you may be on the path to becoming depressed. And the bug's name is Dilister, which is an oral pathogen often associated with periodontal disease. And it's specifically Dilister invisus. And this observation is in step with data that are out there in the literature that argue that poor oral health is a correlate of depression and a possible upstream determinant of our gut microbiota. After all, we are what we eat. And so, as mentioned in our opening, bacteria of the relatively depleted in relation to depressive symptoms were Bacteroides. So if you don't have Bacteroides or Ruminococcus or Holdermanella, you're going to be depressed. Blothia, which was until recently classified under the genus Ruminococcus, is and these others like Roseburia and Proteobacter were also identified as good actors. Now, there were 5 bad actors in addition, where there were 5 bad actors. Dilister is one of them, and one was another Blothia species, and Blothia producta, and a Lactoclostridium, and an Ocealobacter, but they're only associated. But the surprising finding to me was their amplicon sequence analysis also uncovered the genus Bifidobacterium, and it showed a positive correlation with depression. Now, if you go out and you look to your favorite probiotic, a lot of the probiotics have Bifidobacter, specifically Bifidobacter longum, in them. So if you're taking Bifidobacter longum as a supplement, you may be sending yourself on a path to becoming depressed. But we don't have any hard data to show that one way or the other, because Bifidobacter has multiple strain types and the genus is highly diverse. So it's conceivable that the Bifidobacter in the probiotics is innocuous, whereas the Bifidobacter that was associated in their correlation study with depression was a specific strain that the 16S technology couldn't delineate, because all 16S is just reporting 16S. It's not telling about the other metabolites the Bifidobacter may be manifesting, and some of them may be neuroactive in causing depression. So summing up, in their over 3,000 study subject analysis using this ethnically diverse population, they demonstrate a robust association between the gut microbiota and depressive symptoms. Now, these associations were observed largely invariant across these ethnic groups and withstood adjustment for a uniquely large set of relevant confounders. And they ended up with this, if you will, call to arms, where they're calling the scientific community, everyone from neuroscientists to microbiologists to statisticians, to help identify potential targets for psychobiotic interventions that may positively impact depression and the well-being at both an individual and a population level. So it's some Christmas reading for everyone.
I think before I invest in companies that are doing that, I'd like to see a test of whether these particular microbes are in fact causal. So classically, you implant some cohort
of microbes
into a germ-free animal like a mouse, and then ask, does the microbiota impact the behavior?
And in their narrative, they describe some of those studies, Michelle, as they specifically describe a study by Kelly showing the transferring of gut microbiota of depressed human patients to germ-free rats induce behavioral and physiological features characteristic of depression in the recipient animals, suggesting that gut microbiota may be involved in causal pathways leading to depression. So
boy, that's really promising.
These two papers are chock-a-block full of lots of good information. So if you're interested in this field, take a look at these papers and the review articles I dumped in the show notes, because they'll serve as a good background. And again, this is a bridge from microbiology to neuroscience to mental health. And so I think we all have to be talking amongst ourselves to really begin to help these individuals suffering from depression.
That rat study that you described is really promising, because not only does it is it consistent with the idea that the microbes and these small molecules they're producing, neuroactive molecules, may actually be changing behavior, but also it's now an animal model to do some of the studies that you were suggesting, some drug screens, et cetera, to modify that interaction.
And that paper was from 2016, and it was entitled Transferring the Blues, Depression-Associated Gut Microbiota Induces Neurobehavioral Changes in the Rat. And it was in the Journal of Psychiatric Research in 2016. So I'm sure many other studies have been carried out since. So this is a big, rich field. These papers are well referenced. 93 references in this short paper on the Rotterdam study. And the second paper is equally well referenced. So I commend them for your holiday reading.
Thank you, Michael. All right. That's quite something. Yeah. Michelle, what do you have for us?
I have a paper that just became available in MBio, and the title is Purine Nucleotides Interfere with Cyclic Di-AMP Levels and Act as Adjuvants to Resensitize MRSA to beta-lactam antibiotics. And it is the work of a group that is at the University of Galway and a collaboration with the folks at the University of Sheffield in the United Kingdom. And the authors are Aaron Nolan, Merv Zedin, Igor Kavatsky, Christopher Campbell, Lucy Irwin, Rebecca Corrigan, Angelica Grinling, and James O'Gara, the two PIs. So first, let's talk about Staph aureus, Staphylococcus aureus. This is a gram-positive organism that is very common, circulating in human populations. It can, if given the opportunity through some defect or break in our immune system or barriers, it can cause a variety of diseases, including skin infections, pneumonia, urinary tract infections, endocarditis, and then in the worst case scenario, if it gets into the blood, it can cause sepsis and toxic shock syndrome. Not only that, but through the awesome power of horizontal gene transfer, Staph aureus has acquired a number of genetic elements that have endowed it with new properties, and including the ability to resist treatment to beta-lactam antibiotics. So these are penicillin is the one you probably have all heard of. Methicillin is part of that class. And we know that MRSA strains have acquired a mobile element that confers the resistance to methicillin. And that's because it encodes a distinct penicillin binding protein on this 30 kb element that is able to continue to do its job of building a cross-linked peptidoglycan cell wall, even in the presence of beta-lactam antibiotics. Not only that, but after Staph aureus acquired this MECA gene on a genetic island, it later it acquired a pore-forming toxin from a phage, bacteriophage, that infected the strain. And then later still, it required another large island by horizontal gene transfer that equips it now to survive on our skin. So rather than be relegated to our nasal passages, now these strains that have this new acme island are able to tolerate the antimicrobial peptides and low pH of our skin, and therefore these strains that are now methicillin resistant, have a pore-forming toxin, can live on our skin, and guess what? This is why we see cautions in gyms that you might attend, or cautions to athletes to not share towels, to wipe down equipment before you use it, because the sweat from the previous user may have had MRSA, and you could then be exposed. And if you're vulnerable, you could have some serious infection. So not only is this more highly transmissible, but it's also resistant to the whole class of beta-lactam antibiotics. Anybody want to chime in about MRSA in particular? Any favorite stories?
It can remain on those gym surfaces for upwards of 365 days. So that means a year. I mean, it's a very hardy microorganism. It has baked in suntan lotion in its pigments. So it's resistant to the effects of ultraviolet light. I mean, staph came loaded for bear with virulence factors. And these are just the ones that protected in the environment. When it gets into our body, it's got a panoply of virulence factors that just get expressed, and it fights a battle with our immune system. The biggest one is it binds our immunoglobulins and takes it out. Many of us in lab have used something called IgZorb, which is, of course, a staphylococcal product.
But we digress.
We digress. You asked.
So not only is MRSA a problem, but also we want to be able to continue to use our beta-lactam antibiotics. These are the safest and most effective class of antibiotics we've got. And so this group and others are really motivated to try to understand how can we basically resuscitate. Can we come up with an adjuvant, which is the clinical term we use for another ingredient we can add when we add a methicillin to a therapeutic that can make the methicillin more effective. So the other background is that it's been observed that when staph acquired resistance to methicillin, they also acquired a few other traits, including more apt to have a different cell size, biofilm formation, osmoregulation. They had higher levels of signaling molecules that coordinate those traits, including PPGPP, which is classically known as magic spot, and also cyclic di-AMP. Both of these small messengers coordinate major transcriptional changes and can change the cell type of a bacterium, so they can switch from one mode to another depending on what the local conditions are like. Now, what PPGPP and cyclic di-AMP have in common is they're both made from nucleotides and nucleosides. So this is the heart of the paper. They're looking at the interaction between these nucleoside pools and the resistance to beta-lactam antibiotics. So the paper starts by just doing a simple experiment and asking whether addition of guanicides will change the sensitivity of Staph aureus to beta-lactam antibiotics. So if, in Table 1, for example, they look at their strain and find that a certain concentration of oxacillin, their beta-lactam of choice, has a minimal inhibitory concentration of 64 units. But if they add small quantities of guanosine, this nucleoside, now the amount of oxacillin that you need to add to inhibit the concentration drops from 64 down to around 2 to 4. And as you increase the amount of guanosine that you expose the microbes to, the amount of oxacillin required to inhibit growth drops even further.
So Michelle, does that work for the other purine, adenine?
It doesn't. Adenine actually has the opposite effect. So if you add adenine, you see that you require extra amount of oxacillin antibiotic to stop growth. So clearly they've got some fascinating bacterial responses here, and it's worth digging in. So what they do, fortunately for us, is they want to first see, is this just unique to their particular strain, or is it more common to other MRSA strains out that are circulating in the human population, including the dreadful USA 300 strain, which is endemic in some areas. And unfortunately, they find the very same response for these other clinical isolates, that addition of guanosine drops down the amount of oxacillin that you need to add in order to stop the growth. So that's great news. So now they want to understand how this works and what cellular pathways might be involved in this collaboration, this adjuvant activity between nucleosides and the beta-lactam antibiotics. So they turn to genetics. And fortunately, we have a large literature on the biosynthetic pathways for these nucleosides and also scavenging nucleosides from extracellular sources. And they provide us in Figure 1 with a really clean schematic that's got the components laid out so that you can tell where they function in the cell, and shows the pathway, and it's got the gene names, so that as we read the paper and look at the different figures, we can refer back to this figure and see just what, envision just what pathway they're manipulating. So they're able to take advantage of a community resource called the Nebraska Transposon Library, where the Staph aureus field has made available to all their community members this large transposon library where they know where each transposon is. They also know from the literature, which genes are important for nucleoside salvaging from the environment or biosynthesis. So now they can sit and make some really logical guesses. If this is nucleoside uptake, if it's nucleoside biosynthesis, then loss of function mutations in particular genes should impact this sensitivity to oxacillin. So they do those experiments, and they show us some quantitative data in Table 1, but more impressively, they, at least to me, they also were able to use another very elegant technique, which is a disc diffusion assay, to ask whether the adjuvant activity was specific to this one antibiotic they tried, oxacillin, or is it instead true of the other beta-lactam antibiotics. So in this disc diffusion assay, you basically smear your strain of interest on the agar plate and then put tiny little discs that are impregnated with different antibiotics on the plate and then incubate. So if the bacteria can grow right up to the disc, then they are clearly resistant. But if you see a zone of clearance, and the size of that zone of clearance tells you just how sensitive they are to that particular antibiotic. So really lovely data shown in Figure 2A is this disc diffusion assay. And Vincent, one of our authors described the experience of looking at this plate after it'd been in the incubator. Do you want to share that?
Yeah, this is from Merv, who is a Cypriot scientist. To tell you a little bit about her career in a bit, but Merv points out that the highlight of this project was imaging. Sorry, I got the wrong person here. Yeah, I'm talking about Aaron. Aaron is the other collaborator on this paper.
A second-year graduate student. Yeah.
Second-year graduate student. Sorry, Merv. Whose project memory, the summer morning I first observed the dramatic effect of guanosine on MRSA beta-lactam resistance on disc diffusion plates, which is the assay that Michelle is telling us about.
When you look at that disc diffusion assay, you can't think of it as a plaque assay.
I can't?
No, you can't. It looks like a plaque assay to you, but it's not. It's
Okay.
not a plaque assay.
It's very pretty, though.
It's very pretty, but I was expecting you to call it a plaque assay.
No, I've seen them before. I've actually done some years ago.
Yes.
Wow. So they could also use this diffusion assay to say, all right, we've seen this fantastic interaction between the guanosine and beta-lactam antibiotics. What about other antibiotics? So they do the same experiment, but now they use tetracycline or vancomycin, chloramphenicol, gentamicin, which are antibiotics of different chemical classes that have different cellular targets. And here they see no impact of these nucleosides. So they can conclude that it is in general true of the beta-lactam class, but it's not a widespread resistance to different types of antibiotics. So really, really lovely data that would certainly motivate me to keep going. They also wanted some quantitative data, just what is the extent of growth inhibition or killing. And so they quantify colony-forming units per mil of broth over a 24-hour period. And they saw actually an increase in 2 logs of killing when they added guanosine to a standard oxacillin concentration. So imagine that just by adding this nucleoside, you get a 100-fold increase in killing of your target. And what's especially encouraging is the concentrations they're using throughout this paper are actually in the therapeutic range that are currently being used for humans. So this is not some kind of far out proposition. This really looks promising.
Will they be able to compound the guanosine with the oxacillin derivatives and put it into the same pill?
So that's a question for a chemist, and I could not answer that. But before they even do that, they want to really make sure that they understand this. And to these authors' credit, they use a number of independent approaches to make sure they really understand what's happening in these cells. In fact, when they do a growth curve, one thing they're noticing is that when they add guanosine in the absence of the antibiotic, there's an apparent increase in cell growth, culture growth, based on the optical density of the culture. Yet, when they plate for colony-forming units, how many viable cells are in that culture, they see no difference. So there's this paradox between the optical density and the colony-forming unit. So what they do, as any well-trained microbiologist would do, is they actually look at the cells, take them out of the culture, and take a look under the microscope. And what they discovered was pretty striking. It is shown in figure 5A and B that when they add a little bit of guanosine, the Staph aureus cells get a little bigger. If they add oxacillin by itself, they get even bigger still. And if they add the two together, the cells look really quite large and swollen. So they've got another simple assay where they're convinced that there is an impact, an interaction between the nucleoside and the beta-lactam antibiotic. So now they want to understand what's going on. And here's where they leverage the transposon library that's available in the community. So they know from the literature what the genes are that are required for making the guanosine or scavenging it from the environment. So they can compare side by side strains that are capable of making or scavenging the nucleoside or strains that aren't. And then they expose them to the oxacillin. And what they find is loss of function mutations in the biosynthetic genes, which are called PER, increased resistance to oxacillin and other beta-lactam antibiotics, as did loss of function mutations in any of the transporter genes that are used to scavenge the nucleosides. In contrast, if they looked at a strain that lacked a repressor of biosynthesis of purines, they saw the opposite effect. Now they are more sensitive to the antibiotic. So by this approach, they are able to do a number of experiments and complementation tests that I won't go into that allowed them to assign functions using these really simple assays, sensitivity to oxacillin. So with that as their foundation, they then wanted to look in more detail at what is happening in these cells. And in particular, is it an impact on cyclic AMP, cyclic di-AMP, which, as I mentioned, they knew from the literature is associated with osmoregulation, or resistance to osmotic stress, cell shape, and also has some not yet understood impact on resistance to methicillin. So one approach they took to look at this in more detail was a classical genetics approach, where they looked for suppressors of this phenotype, suppressors of the capacity of guanosine to augment the antibiotic oxacillin. So to do this, they passaged their MRSA strain in media supplemented with both oxacillin and guanosine multiple times, and they eventually were able to isolate 16 rare spontaneous mutants that were able to survive the combination of oxacillin and guanosine. So then they could verify that, in fact, yes, their minimal inhibitory concentration had increased due to the addition of guanosine. And then they could use whole genome sequencing and ask, what changed? Where were the spontaneous mutations? And what can they tell us about the mechanism of this interaction? And one of the genes that they identified through their suppressor analysis was a gene they studied earlier, NUPG, which is a major transporter for the biosynthesis of guanosine. So again, this is very encouraging. They're using a different approach, and the cells are telling them, yes, 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 the nucleoside pools are important. And then they isolated mutations in a number of other pathways, in general, related some sigma factors related to the stress response, or tRNAs related to translation, and that's work for another day. So let's now focus on what is happening here with the nucleoside supplementation. Is this an effect on GTP, PPGPP, or the cyclic di-AMP? So fortunately, there are biochemical techniques that can allow you to quantify each of those. And when they first looked at GTP, they saw really nothing dramatic in the presence of guanosine with or without the oxacillin. Likewise, when they used thin layer chromatography to look at PPGPP, there was really nothing striking, nothing you'd want to chase down. But when they used an ELISA assay, which allowed them to quantify the amount of this messenger molecule cyclic di-AMP using antibody, they saw that when there were loss of function mutations in either transporters or an enzyme that phosphorylates the nucleoside once it's in to trap it in the cytoplasm so the cell can use it. In both cases, they saw a decline in the cyclic di-AMP pools, which again shows that there is a connection between the transport and scavenge and biosynthesis of these guanosine building blocks and the pools of these second messengers, including, or in particular, cyclic di-AMP. And that was consistent with their observation, because the literature had also said changes in cyclic di-AMP does affect cell size and also osmotic resistance.
So the one question I have that you probably don't have an answer for, but Pseudomonas can be resistant to methicillin or beta-lactam classes of antibiotics. And Pseudomonas is notorious for not bringing in any preformed anything. It breaks everything down to the basic building blocks. So I'm wondering if the guanosine would have the same effect in a microbe like Pseudomonas that literally doesn't bring anything in preformed.
Yeah, it might be harder to get the nucleoside in if they don't have that transporter ready to exploit. Yeah, but I'm sure they will be very eager to test this against some other of our dreadful
other pathogens that are significant,
antimicrobial resistant,
where you want a really strong bactericidal antibiotic to knock down the infection.
So they did one more genetic experiment that showed that if a cell lacks an enzyme that breaks down cyclic di-AMP, then they actually increased resistance to beta-lactam. So these people really know their genetics, they know their signaling pathways, and they were able to establish very clearly that the nucleoside pools were impacting sensitivity to oxacillin. And again, they see these impacts in concentration ranges that are used currently in the clinic for a number of treatments. And in fact, they point out in their discussion that there are a number of cancers and viral infections that are already being treated with derivatives of different purine nucleotides. So they're optimistic that once we better understand how this is working and understand how to formulate them and what maybe off-stream effects you have, we have potential to augment the treatment of infections with MRSA and other bacterial pathogens by beta-lactams, by also including as an adjuvant a derivative of a nucleoside. So this is really hopeful that we have a new, clever approach to take advantage of what's known about metabolism and signaling in these bugs, and hopefully resuscitate the beta-lactam classes of antibiotics.
Let me tell you more about the two first authors on this paper. Merv is a Cypriot scientist who loves microbiology, went to Imperial College for her BSc, stayed there for her PhD with Professor Angelika Grundling, where she studied the requirement of cyclic di-AMP for growth of S. aureus. They then moved to the O'Gara lab at the University of Galway to integrate molecular microbiology expertise with antimicrobial resistance research. And Merv has worked with Aaron, and she really has enjoyed that and has had a lot of grateful discussions and motivations, and also other people in the lab. And her project highlight was imaging the impact of oxacillin and guanosine on the size of Staph aureus cells by confocal microscopy. She writes, our WhatsApp group chat was busy that day, and we may have had a pint of Guinness to celebrate.
I hope you did, Merv.
That's figure 5 for those of you following along. Again, an open access paper.
Mm-hmm.
And Merv's pastimes include cold water swimming in the Atlantic Ocean, even in winter. Oh my gosh, yoga and hiking. And her advice for you is follow your passion, do what you enjoy, and find the right people to support you throughout your career. That's great. You do need to find the right people.
Isn't the water off of Galway warm because of the Gulf Stream?
I have no idea.
We'll have to go and test it.
Yeah, right. And Aaron is a second-year grad student who's transitioned from an undergraduate degree in biopharmaceutical chemistry to a master's in biotechnology and now a PhD in the O'Gara lab. Aaron's fascinated by antibiotic resistance and metabolism, and they bring together his interests in chemistry and biology. Aaron is grateful to be part of an ambitious research group and to benefit from the support and knowledge of supervisors Merv and Jim. And as I told you, his project memory with the size of the clearings on the disc diffusion plates. His pastimes include sports, reading, and brewing beer with the Microbiology Society in Galway.
All right.
And Aaron's advice for you is to do what is best for you, follow your interests. I'm living the maxim that you'll never work a day in your life if you do something you love. I love it. That's my maxim as well. You won't call it work.
Congratulations to the two of them on this project. They're off to a great start, and their collaborators. Yeah.
Thank you, Michelle. That'll do it for TWiM 278. The show notes are at microbe.tv/TWiM. Send your questions and comments to twim@microbe.tv. If you enjoy our work, please consider supporting us, microbe.tv/contribute. Michelle Swanson's at the University of Michigan. Thanks, Michelle.
Thank you.
Michael Schmidt's at the Medical University of South Carolina. Thanks, Michael.
Thanks, Vincent. Thanks, Michelle.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.